Morning, ma'am.
Hello, hello. How are you all? How is the winter season going on? Chilled? Okay, so let's take a break from this chilled atmosphere.
Good
Chikori classes joined. Have you all, both of you joined Chikori
morning,
class?
ma'am. Good morning, ma'am.
Good morning, good morning. Yes, class 21. Who is there? Class 21 Chikori? Only one girl I can see. 22? Only 3, 4 girls. 3, 4, no, Allah, only 2. Why? They'll come? 21, 22 Chikodi will, they will come, other people will come.
Sir, ma'am, ಬರ್ತಾ रहेगा.
What, you had a break or what?
Yes, ma'am.
एक ब्रेक इतना और
Sorry, okay, okay. Others ಮತ್ತೆ ಯಾರಿದ್ದಾರೆ ಅಂದರೆ we have only Ranebennur and Chikkodi. Dharwad, we have Dharwad. Okay, fine. Anybody remembers what we were doing? What we were doing? What we were studying? Anybody remembers? No? Very good. ಅಟ್ಲೀಸ್ಟ್ ಯಾವ ಚಾಪ್ಟರ್ ಮಾಡ್ತಾ ಇದ್ದೇವೆ ಅದನ್ನ ಹೇಳ್ರು ಸ್ವಲ್ಪ ಸಮಾಧಾನ
Biotechnology.
ಸ್ವಲ್ಪ ಸಮಾಧಾನ ಮಾಡ್ಕೋತೀನಿ ವಿ ವರ್ ಡೂಯಿಂಗ್ ಬಯೋಟೆಕ್ ಓಕೆ ಸೊ ಇಫ್ ಯು ರಿಕಾಲ್ we have done restriction enzymes ವರ್ಗೂ we have completed till restriction enzymes and we had to start with the properties of ideal vector and gel electrophoresis separation and isolation. Open your books, come on, revise. You have forgotten everything. One vara gap ಆದರೆ ಈ ಥರ ಆಗತ್ತೆ. You don't remember only. ನೆನಪಾಯ್ತು? We had done restriction enzymes, nomenclature of restriction enzymes, then how this restriction enzymes are used to cut the DNA, make a recombinant plasmid, insert into the host cell and make a many copies. Till this we have completed. Ranebennur girls, settle down quickly, open your textbooks and start paying attention. Okay. So moving ahead, your next concept was about this. Gel electrophoresis. Right? Okay. So what is gel electrophoresis
Yes,
or
ma'am.
why it is needed? What is a purpose or intention ಏನು what do we do using gel electrophoresis? Anyone?
To find the gene which
No, not to find the gene. Think. ಯಾಕೆ ಮಾಡ್ತೀವಿ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎನ್ಸೈಮ್ಸ್ ತಗೊಂಡು ವಾಟ್ ಡಿಡ್ ವಿ ಡೂ ಸಿ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎನ್ಸೈಮ್ಸ್ ತಗೊಂಡ್ಬಿಟ್ಟು ವಾಟ್ ವಿಡ್ ವಿ ಡೂ ವಾಟ್ ಡಸ್ ದ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎನ್ಸೈಮ್ ಡೂ cuts the DNA. Restriction enzyme will cut the DNA. Okay, which means we got DNA fragments. What did we do? Please recall, people, we have isolated the gene of interest and we have isolated the plasmid. The ಎರಡು gene of interest and plasmid we have cut using restriction enzyme. Now this DNA what we have isolated, ಈಗ one cell ಇಂದ ಇಷ್ಟುದ್ದ DNA isolate ಮಾಡ್ತೀವಿ. Okay, isolate ಮಾಡಿದ್ಮೇಲೆ we make a cut using what? Using restriction enzyme. Cut ಮಾಡಿದ್ಮೇಲೆ we get many different fragments. These fragments have to be separated. And for separating these fragments, we use the process called as gel electrophoresis. So, ಇಲ್ಲಿವರೆಗೂ ಬಂದಿದ್ವಿ ನಾವು. So now, to again resume with gel electrophoresis is going to help to separate and isolate the piece of DNA For example, if you remember, I had taken insulin gene. Let's say ಇಷ್ಟುದ್ದ DNA ಇದೆ human cell ಇಂದ, ಅಷ್ಟುದ್ದ DNA ನನಗೆ ಬೇಡ. What I want, I only want a small fragment of DNA. So that small fragment of DNA, ಇಷ್ಟೆಲ್ಲ fragments, using restriction enzyme, you got very much of fragments. From amongst all those fragments, I have to select only one small fragment. Correct? So for that, I have to separate all these fragments based upon the molecular weight. Based upon the molecular weight of that fragments. So what is being used? The apparatus called as gel electrophoresis is used, where you can see there is a tray of gel electrophoresis, and then there is a matrix. This matrix is made up of,
Gross gel.
yes, agarose gel, which is obtained from seaweeds. So what happens when the sample sample means what? Sample contains the fragments of DNA. ಈ sample ಅಲ್ಲಿ ಏನಿದೆ, sample has a various fragments of DNA. This DNA fragment sample, when it is mixed with a solution called as, sample should be stained with ethidium bromide. Yes. ಇಲ್ಲಂತಂದರೆ ಡಿ ಎನ್ ಎ ಕಣ್ಣಿಗೆ ಕಾಣಲ್ಲ, ಇಟ್ ಇಸ್ ಟ್ರಾನ್ಸ್ಪರೆಂಟ್ ಅಲ್ಲ, ಯು ಹ್ಯಾವ್ ಟು ಸ್ಟೇನ್ ಇಟ್. ಸೊ ಇಥಿಡಿಯಮ್ ಬ್ರೋಮೈಡ್ ಸ್ಟೇನ್ ಈಸ್ ಯೂಸ್ಡ್ ಆ್ಯಂಡ್ ದರ್ ಈಸ್ ಅ ವೆಲ್ ಹಿಯರ್. ಅಲ್ಲಿ ಆ ಸ್ಯಾಂಪಲನ್ನು ಲೋಡ್ ಮಾಡ್ತಾರೆ ಆ್ಯಂಡ್ ದೆನ್ ಇಟ್ ಈಸ್ ಕನೆಕ್ಟೆಡ್ ಟು ದ ಪವರ್ ಸಪ್ಲೈ. Now, because the DNA is negatively charged, once the power supply is on, the DNA will start, this has to be left for a few hours. Hange itbidbidbeku, okay. After many hours, you can see the movement of DNA from cathode towards the anode. Why? Because DNA is what? DNA is negatively charged, and therefore it will move towards the positive anode. Why DNA is negatively charged? Why DNA is negatively charged?
First,
Yes?
due to
Yeah,
presence of two phosphate group, ma'am.
Yes, correct. Due to the presence of phosphate group, we say DNA is negatively charged. Therefore, when the power is applied to this apparatus, after 3 to 4 hours, you can see DNA based upon its molecular weight. You can see the fragments which are running. So, idana yen ಮಾಡ್ತಾರೆ? This has been seen under autoradiography. So where some orange band can be seen. Autoradiography or also UV light can be also used. Depends. E tray na, there is a chamber. A chamber alli there is a UV light or there is a X-ray light. Adar ಕೆಳಗಡೆ ಆ ಟ್ರೇ ಇಟ್ಬಿಟ್ರೆ you have a monitor. ಇದಕ್ಕೆ ಒಂದು ಮಾನಿಟರ್ ಕನೆಕ್ಟೆಡ್ ಇರುತ್ತೆ. ಆ ಮಾನಿಟರ್ ಮೇಲೆ you can see the bands. So bands which are larger in molecular weight, they will be behind because they are very high in their molecular weight. Therefore movement of band becomes slow, and the bands which are running faster or which are ahead are low molecular weight because their weight is less. Therefore they can easily move forward. So based upon now my gene of interest, let's say I had selected insulin, correct, and insulin 200 base pair irbodu, for example, I'm telling you. So this prior knowledge of the gene will help to isolate. Maybe this is my gene of interest. What I will do, I will cut this piece of DNA and remove that small bulk or the small piece of that DNA from that gel. This step of removing this cut piece of DNA is called as elution. Yes, underline that word, okay? Elution means what? The small piece of gene of interest which is cut from that gel and removed. ಆಮೇಲೆ ಏನು ಮಾಡ್ತಾರೆ? That cut gel ಅದರ ಮೇಲೆ ಡಿ ಎನ್ ಎ ಬ್ಯಾಂಡ್ ಕಾಣ್ತಿರತ್ತೆ. ಯು ಕ್ಯಾನ್ ಸಿ ದ ಡಿ ಎನ್ ಎ ಬ್ಯಾಂಡ್ ಬಿಕಾಸ್ ಯು ವಿ ರೇಸ್ ಅದ್ರ ಮೇಲೆ ಬಿಟ್ಟ ಮೇಲೆ ಇಟ್ ವಿಲ್ ಫ್ಲೋರಸೆಂಟ್ ಆಗಿ ಆರೆಂಜ್ ಥರ ಬ್ಯಾಂಡ್ ಕಾಣುತ್ತೆ. ಸೊ ನಾವು ವಾಟ್ ಯು ವಿಲ್ ಡೂ ಆಫ್ ದಟ್ ಕಟ್ ಡಿ ಎನ್ ಎ, ಯೂಸ್ ದ್ಯಾಟ್ ಸ್ಮಾಲ್ ಪೀಸ್ ಆಫ್ ಡಿ ಎನ್ ಎ, ಇಲ್ಲಿಂದ ಏನು ನಿಮಗೆ ಜೀನ್ ಆಫ್ ಇಂಟ್ರೆಸ್ಟ್ ಸಿಕ್ತಲ್ಲ, this gene of interest is used to ligate with the plasmid vector and ಇದರಿಂದ ಸೇರಿ we make rDNA. So the process of gel electrophoresis is to separate our gene of interest from the mixture of fragments. ಯಾಕಂದರೆ recall what we had did, ನಮಗೆ ಯಾವುದಾದ್ರೂ ಬಯೋಟೆಕ್ನಾಲಜಿ ರಿಕಾಂಬಿನೆಂಟ್ ಡಿ ಎನ್ ಎ ಅಂದರೆ ಏನು, ವಾಟ್ ಇಸ್ ದ ಮೀನಿಂಗ್ ಆಫ್ ಜೆನೆಟಿಕ್ ಇಂಜಿನಿಯರಿಂಗ್. ಹಿಯರ್ ವಿ ವಿಲ್ ಟ್ರೈ ಟು ಟೇಕ್ ಅ ಸ್ಮಾಲ್ ಪೀಸ್ ಆಫ್ ಡಿ ಎನ್ ಎ ಫ್ರಾಮ್ ಅ ಬಿಗ್ ಡಿ ಎನ್ ಎ. ಲೆಟ್ಸ್ ಸೇ ಹ್ಯೂಮನ್ ಸೆಲ್ಲಿಂದ ಒನ್ ಡಿ ಎನ್ ಎ ಇಡೀ ಡಿ ಎನ್ ಎನ ಐಸೋಲೇಟ್ ಮಾಡ್ತೀವಿ, ಬಟ್ ವಿ ಡೋಂಟ್ ವಾಂಟ್ ಹೋಲ್ ಡಿ ಎನ್ ಎ, ಐ ಜಸ್ಟ್ ವಾಂಟ್ ಅ ಸ್ಮಾಲ್ ಪೀಸ್. ಆವಾಗ ವಿಲ್ ಯೂಸ್ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎನ್ಸೈಮ್ ಟು ಕಟ್ ದ ಡಿ ಎನ್ ಎ. ಸೊ ಕಟ್ ಮಾಡಿದ ಮೇಲೆ ಐ ಜಸ್ಟ್ ವಾಂಟ್ ಓನ್ಲಿ ಒನ್ ಪೀಸ್ ಆಫ್ ದಟ್ ಡಿ ಎನ್ ಎ. ಇನ್ ಮಿಕ್ಕಿದ ಪೀಸಿಂದ ಐ ಹ್ಯಾವ್ ಟು ಸೆಪ್ರೇಟ್ ದಟ್ ಪೀಸ್. ಫಾರ್ ಸೆಪ್ರೇಟಿಂಗ್ ದಟ್ ಪೀಸ್ ವಿ ವಿಲ್ ಯೂಸ್ gel electrophoresis, and here that separated fragment would be cut from that gel and removed out. After that what we will do amplification, that is multiple copies, PCR anta ide, that is there in the next step, we will see that later. Copies anna multiply ಮಾಡ್ತೀವಿ. ಯಾಕೆ? because there is possibility that you may fail the experiment. ಈಗ ಫೇಲ್ ಆಯ್ತದಂದ್ರೆ you cannot go again back to step 1. So that's why we keep many samples. ಒಂದೇ ಕಾಪಿದು multiple xerox copies ಮಾಡಿಡ್ತೀವಿ ನಾವು, just for a safer side. And then whatever gene we isolated, that we ligate with a plasmid vector and form a recombinant DNA. This is the same step that is shown in your book also. If you see restriction enzyme ಕೆಳಗಡೆ, last time I had told you sticky ends will get joined and forms a recombinant DNA and then put that recombinant DNA into host. That is the same step what we are doing here. Getting? So gel electrophoresis is a technique for separating the DNA fragment, and the principle of gel electrophoresis is based upon the molecular weight. High molecular weight would be running slow, low molecular weight would little bit run faster. Clear till here? Anyone has any doubts? Do this. Let me see how much you have understood.
Option A. Option A. A.
Option A.
Everybody, separation and isolation, construction of recombinant DNA by joining. What enzyme do we use for joining people?
Ligase.
Yes, okay. Culturing the host cells in medium. Okay, yes, yes. A is more correct. What do we tell about this?
Option B.
Option B. Size only.
Size and charge. In agar of gel electrophoresis, DNA molecules are separated on the basis of what? On the basis of charge, on the basis of size, on the basis of charge and size, all of this. Charge and size ಎರಡು ಆಗಲ್ವಾ? What do you think? Or it is only size? Okay, most of you are going with size. Why not charge?
DNA fragments are negatively charged.
Correct, because all DNA molecules are negatively charged. So charge is not going to play any important role here. Ellaru negative charge ide, ellaru will move, all of them will move. But movement yaud fast ide and yaud slow ide, which is going to move faster and which is going to move slower, will be decided by the size. Therefore, you get this as a more right answer. Good job, people. Quick, direct answer.
The option C.
C.
Yes,
Option
option
C.
C. Direct answer, yes. Good going. See, you can see the bands, so beautifully bands.
Option A.
Option E.
Option
A.
A. Option A.
A.
Option E.
Read properly. Natural polymer obtained from seaweeds,
Option
natural
A, ma'am.
polymer obtained from fungi.
Option A, ma'am.
Option A, ma'am.
Okay, fine. Direct answer.
Option B.
D. Option B. Option B.
Yes,
Option B, ma'am.
yes.
Option B.
A yake? Band pattern, not A, B. They're asking about band pattern. Okay, please give the polls, raise your cue cards. Raise your cue
Ma'am,
cards,
zoom in.
people. Hold second, don't answer, wait, people wait. Let us see
Ma'am, zoom.
responses. Poll is active, you can raise your cards. Ranemanur girls, raise your cards.
Ma'am, zoom the picture.
Zoom, okay.
Option A.
Wait, visible?
Yes, ma'am.
Okay, Ranemanur girls, class 2, who is with you?
Option A, ma'am.
Then A.
Wait. Chikodi class 20, where are your answers? Chikodi class 20,
Coordinator
your
is
answers.
Coordinator ಹೊರಗೆ ಹೋಗಿದ್ದಾರೆ.
Not audible, a little bit louder.
Coordinator is outside.
Okay, class 21, Chikodi class 21. Okay. Okay, I have got A as maximum option. Okay. Which is the criteria for movement of DNA fragment on agarose gel? Smaller the fragment size, the farther it moves. Positively charged fragment move to farther end. Negatively charged fragments do not move. The larger the fragment size, the farther they move. So A is most appropriate. Agree? Positively charged fragments, we do not have
Yes,
positively
ma'am.
charged fragments. Negatively charged fragments do not move. No, they do move. Every molecule will move. The larger the fragment size, the farther it moves. No, it's ulta. Right? Okay. Direct answer.
Option B.
Option D.
Option B.
Dharwad,
D.
come on,
Option
baby.
D. D.
Option D.
Option A, ma'am.
D.
Option fourth.
Option D.
4.
Fourth.
Cutting of DNA fragment and extracting them from gel. And A ಏನದೆ cutting of DNA by restriction. Okay.
Option D, ma'am.
Hmm.
Option B, ma'am.
What will you answer to here? Separation of DNA fragment using agar. ಇಲ್ಲಿ ಏನು ಆನ್ಸರ್ ಮಾಡ್ತೀರಾ for B? Gel electrophoresis, correct? B ಇತ್ತಂದ್ರೆ this should be gel electrophoresis. This is wrong. Okay, yes, I would also go with D. Fine. Okay, assertion. No, ಇಲ್ಲ assertion ಅಲ್ಲ. We have a statement based question. Come on, raise your cards, people.
Ma'am, option C. Option C,
Okay.
ma'am. Option C.
Dharwad strength is less today. There is no boy only. Yakro yellogidare? Where are boys today?
Monday is preparatory exam, ma'am.
Oh, howda? From college or boards?
College, ma'am.
ಕ್ಲಾಸಿಗೆ ಅಟೆಂಡ್ ಮಾಡಲಾರ್ದೆ ಇವ್ರು ಪ್ರಿಪೇಟ್ರಿಗೆ ಏನು ಪ್ರಿಪೇರ್ ಆಗ್ತಾರೋ ನೋಡೋಣ. ಓಕೆ ಫೈನ್, ಇಟ್ಸ್ ಓಕೆ, ಲೆಟ್ಸ್ ಸಿ. ಹಾಂ, ಓಕೆ, ವಾಟ್ ಡು ವಿ ಹ್ಯಾವ್ ಆನ್ಸರ್? ಆನ್ಸರ್ ಸಿ. ಮ್ಯಾಕ್ಸಿಮಮ್ ಆಫ್ ಯು ಗಿವಿಂಗ್ ಮಿ ಸಿ ಆನ್ಸರ್. ಏನು ಬೋತ್ ಸ್ಟೇಟ್ಮೆಂಟ್ ಆರ್ ಕರೆಕ್ಟಾ? ಓಕೆ. Gel electrophoresis and elution are two important process in isolation. Okay, okay, agreed. DNA fragments are visualized only after staining with, okay, okay, yes. Okay, a tricky question. Let me see who can crack this. Come on, people. Restriction enzymes are used to cut what? Try, discuss with me only if you want.
Option B.
Option B, ma'am.
Option B, ma'am.
Option B, ma'am.
Okay, double stranded, single RNA into alve alva, right? D and A you don't consider. Okay, double stranded and single stranded, of course double stranded, yes.
Option B, ma'am.
Okay, now restriction enzymes ಮೇಲೆ ಕ್ವಶನ್ ಆಗಿರಲಿಲ್ಲ ಲಾಸ್ಟ್ ಟೈಮ್, ಅದಕ್ಕೆ ಹಾಕಿದ್ದೀನಿ ನಾನು. Restriction enzymes are endonuclease which cleave DNA at specific sites.
Option A, ma'am.
Make
Option A.
DNA complementary to an existing joint. DNA fragments are required in vectorless direct. Okay, okay. A.
Option A, ma'am.
A. All of you A?
Yes, ma'am.
Yes.
Yes, ma'am.
Okay. Good. This is very direct question. Directly from your book. Try. Come on.
Option B.
Option B.
Option B, ma'am. Option B.
B. 1 eco R1, 2 DNA ligase. 1 idu 1 eco R1, 2. Okay. Yes.
B, ma'am.
1 ಯಾಕೆ
Option B,
ಆಗ್ಲಿಕ್ಕಿಲ್ಲ?
ma'am.
1 ಹಿಂಡ್ 2 ಇಲ್ಲ.
Enzymes use
Yes, eco R1 ಯಾಕಂದ್ರೆ because this site GAATTC, this is particular recognition site for eco R1 only. This is a recognition. This is a recognition site for eco R1. Yes. Good. Be careful now and raise the cue cards. Come on, waiting for your responses.
Option C, ma'am.
Option C.
C. Okay. In recombinant DNA technology, the vector DNA and foreign DNA are cut and joined to form recombinant DNA. They are cut by the same restriction enzyme, okay? Which of the following is incorrect? They are asking what is incorrect. You are telling me option C. Most of you are going with option C. Vector and source DNA, when cut by different restriction enzyme, Recombinant DNA can be more easily formed. Incorrect. This is false. But others are true. Vector DNA and source DNA are cut by the same restriction enzyme. True. When cut by the same restriction enzyme, DNA fragments have same kind of sticky ends. True. Vector and source DNA are joined by true. Good job, people. Good one. Good. Okay, PBR. Okay, this is about plasmid. First plasmid ಬಗ್ಗೆ ಓದೋಣ and then we'll solve the next questions. Yes. So what is the next concept? What are the ideal? How should be the ideal plasmid? Or what are the criteria for an ideal plasmid? Correct? That is what your next is. So very first thing what an ideal plasmid should have is, why you require oocyte? What is the purpose of oocyte? Oocyte
To
ಅಂದ್ರೆ ಏನು?
initiate
Yes, again,
to
Replication.
again.
Replications start from origin.
Correct. That is, oocyte is origin of replication. So plasmids ಅಂದ್ರೆ ಏನು? Again going back to basic. ಇದೆಲ್ಲ ಹೇಳಾಗಿದೆ, molecular inheritance ಅಲ್ಲೂ ಹೇಳಿದ್ದೆ. Starting of this topic also I told you, plasmids or bacteriophages. These are what? They are self-replicating or autonomously replicating molecule. They can make its own copy. There are 5 to 100 copies per cell. ಒಂದೇ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಸೆಲ್ಲಲ್ಲಿ so many 100 copies of plasmid can be present. Why? Because they can make on its own replication. Why? Again, because of the presence of oocyte. So ಯಾವುದು vector, if any vector we have to use, not only plasmid but think of any vector should carry oocyte, because ಆ ವೆಕ್ಟರ್ ಜೊತೆಗೆ when you ligate our gene of interest, oocyte ಇದ್ರೆ we can make multiple copies. And when the plasmids are making multiple copies, along with that our gene of interest will also start multiplying. Therefore the presence of oocyte is important. Then second criteria ಏನು?
Cloning site. Selectable marker.
Selectable marker. Now coming to your PBR 322. Yellow itadu. Let me just check. Okay. This diagram. Okay, just a second. You can observe in your books also, it's the same diagram. So what happens in this? The PBR 322 is a artificially made plasmid. Who did this? Nobody knows?
Bolivar and Rodriguez.
Yes. By Bolivar and Rodriguez. So in the Franklin Institute, the Bolivar and Rodriguez had constructed this artificial ಯಾಕೆ ಇಲ್ಲಿವರೆಗೂ we talk about natural vector. Here we talk about natural vector. Natural vector means naturally occurring plasmid. But naturally occurring plasmid ಗೆ there is a only one, most of the time there is only one cloning site. ಸೊ ಒಂದೇ ಕ್ಲೋನಿಂಗ್ ಸೈಟ್ ಇತ್ತಂತಂದರೆ ಯಾವ ಕ್ಲೋನಿಂಗ್ ಸೈಟ್ ಇದೆ ಅದೇ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಜೈಮ್ ಯೂಸ್ ಮಾಡಬೇಕು. ಲೆಟ್ಸ್ ಸೇ ದೆರ್ ಇಸ್ ಓನ್ಲಿ ಹಿಂಡ್ ಒನ್, ದೆನ್ ಹಿಂಡ್ ಓನ್ಲಿ ಹ್ಯಾಸ್ ಟು ಬಿ ಯೂಸ್ಡ್ ಎಸ್ ಅ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎಂಜೈಮ್. ಇಫ್ ದೆರ್ ಇಸ್ ಇಕೋ ಆರ್ ಒನ್, ದೆನ್ ಇಕೋ ಆರ್ ಒನ್ ಓನ್ಲಿ ಹ್ಯಾಸ್ ಟು ಬಿ ಯೂಸ್ಡ್. ದ ಡಿಸ್ಅಡ್ವಾಂಟೇಜ್ ವಾಸ್ ವಾಟ್? ನ್ಯಾಚುರಲ್ ವೆಕ್ಟರ್ ಕುಡ್ ಕ್ಯಾರಿ ಓನ್ಲಿ ಸ್ಮಾಲ್ ಪೀಸ್ ಆಫ್ DNA. Therefore, a artificially constructed vector was made, the plasmid by vector by Bolivar and Rodriguez. The speciality of this vector is what? Here along with ori site adu idde ide, we have ori site. It also has different cloning sites. You can see 1 2 3 4 5 6 7 8. There are almost 8 cloning sites. Cloning or restriction site ಏನಾದರೂ ಅನ್ಬೋದು, cloning or restriction site. If you remember, while teaching you restriction enzyme, I told you name of restriction enzyme and name of cloning site always same. If the name of restriction enzyme is EcoR1, the name of cloning site is also EcoR1. So idu enzyme ide and this is restriction site. Let us write it as a RE, restriction enzyme, RS, restriction site. What happened to this? What happened to this? So whatever restriction enzyme you are using, same restriction sites can be used. EcoR1, EcoR1, BAM-H1, BAM-H1. Enzyme name and restriction site name is same. So first, what does the vector should have? It has ori site. Then the marker here means we are talking about the resistance gene. So what is the meaning of resistance gene now? Selectable marker like antibiotic resistance gene. Why? Because this marker will help to select the recombinant and non-recombinant colonies, or this is also called as transformant. Recombinant ಗೆ ನಾವು transformant ಅಂತ ಹೇಳಬಹುದು. Same, both are same. Transformant or non-transformant. So selectable marker actually helps to select between transformant and non-transformant. Now what is transformant? If you recall, plasmid ಜೊತೆಗೆ if I put my gene of interest, ಇದಕ್ಕೆ rDNA ಅಂತ ಹೇಳ್ತೀವಿ. Now when this rDNA is inserted into the host, ಒಂದು ಯಾವುದೋ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಒಳಗಡೆ, if you have put that plasmid, now if this plasmid has surely went inside the cell or not, ಅದನ್ನ ಚೆಕ್ ಮಾಡಬೇಕು. ಅದು ಹೆಂಗೆ ಗೊತ್ತಾಗಬೇಕು? That would be helped by this selectable marker. Markers are going to help. How they will help? Now we can understand by using this. So how the markers will help? For example, let's say I have used or I have put a particular gene of interest, a small DNA fragment, into this BAM-H1. BAM-H1 is what? It's a cloning site. Ranamanur girls class 1, is it very funny? Or is it very much clear to you? Then you only teach me, it's better. This is not an easy chapter, people. ಇವಾಗ ನಿಮಗೆಲ್ಲ ಮಜಾ ಅನಿಸ್ತಿರ್ಬೇಕು, ಅಥವಾ you must be feeling it is very boring. But I swear to you, this is one of the challenging chapters. If you do not pay attention, your loss, you will not understand it. It's not so easy. Anyways, okay, coming back. So what are we doing? We are putting our small gene of interest into this BAM-H1. BAM-H1 ಏನು? Quick answers. What is BAM-H1? Cloning site or restriction site. So EcoR1 is also cloning site, BAM-H1 is also cloning site. ಆ cloning site ಗೆ ಅದೇ restriction enzyme use ಮಾಡಬೇಕು. I had told this. Restriction enzyme and restriction site, both the names are same, so that you will not get confused. The enzyme name and site name is same. Suppose ಇಷ್ಟೆಲ್ಲ ಪ್ಲಾಸ್ಮಿಡ್ ಇದೆ. ಈ ಪ್ಲಾಸ್ಮಿಡ್ ಅಲ್ಲಿ you can see this much part is ori. Then This orange part is what? They are ampicillin and tetracyclic resistant gene. Resistant gene andre, what will happen if this plasmid is kept in a antibiotic of ampicillin? Because this plasmid has a resistance power, the antibiotic will not kill the plasmid. Instead, it will grow. ಅದಕ್ಕೆ ರೆಸಿಸ್ಟೆಂಟ್ ಜೀನ್ ಅಂತ ಹೇಳ್ತೀವಿ. ರೆಸಿಸ್ಟೆನ್ಸ್ ಮೀನ್ಸ್ ವಾಟ್? The antibiotic will not be able to kill this plasmid. Antibiotics, you know now, you have done human health and diseases, people. Don't sit like dumb. Answer. Similarly, even the tetracycline antibiotic, ಇದೇ ಥರ ಬೇರೆ ಬೇರೆ ಆಂಟಿಬಯೋಟಿಕ್ ಇರುತ್ತೆ ನ್ಯಾಚುರಲಿ ಇನ್ ದ ಪ್ಲಾಸ್ಮಿಡ್. ದೆರ್ ಆರ್ ವೆರೈಟಿ ಆಫ್ ಆಂಟಿಬಯೋಟಿಕ್ಸ್ ಜೀನ್ ಪ್ರೆಸೆಂಟ್ ಆನ್ ಪ್ಲಾಸ್ಮಿಡ್. It can be canamycin, it can be azithro, anything. ಬೇರೆ ಬೇರೆ ಆಂಟಿಬಯೋಟಿಕ್ಗೆ ರೆಸಿಸ್ಟೆನ್ಸ್ ಮಾಡೋ ಜೀನ್ ಪ್ರತಿಯೊಂದು ಪ್ಲಾಸ್ಮಿಡ್ನಲ್ಲಿ ಇರುತ್ತೆ. ದಟ್ಸ್ ಅ ನ್ಯಾಚುರಲಿ. ವೈ? ಸೊ ದಟ್ ಇಟ್ ಕ್ಯಾನ್ ಪ್ರೊಟೆಕ್ಟ್ ದ ಬ್ಯಾಕ್ಟೀರಿಯಾ. It is given to give a natural defense to the bacteria. ಅದೇ ಪ್ರಾಪರ್ಟಿನ ಇನ್ biotechnology what we do simply, where is that gene present? At the same spot I will put gene of interest, or you can say desired gene. So if I put my desired gene at this location, what will happen? Tetracycline resistant gene gets disrupted, which means now if I grow this on tetracycline medium, there would be no growth of the bacteria, no growth of the colonies. Yake? Because tetracycline resistant gene got lost and the cells became sensitive. Sensitive agide andre they have become died, all the bacterial cells died. Ade innond case alli, suppose this I grow on ampicillin containing medium, 2 times plate ಮಾಡಬೇಕು. You have to do this procedure at the same time. Same sample anna grow it on 2 different plates. One plate which contains ampicillin, one more plate which contains tetracycline. These two are antibiotics. So if you observe the ampicillin containing medium, suppose this is a petri plate, adar mele you are seeing the 4 colonies. After one day you can see the 4 colonies. So ampicillin medium alli 4 colonies grow agatte. Let's say for example, because this ampicillin gene is intact, ಈ ಆಂಪಿಸಿಲಿನ್ ಜೀನಿಗೆ ನಾವು ಏನೂ ಮಾಡಿಲ್ಲ, it is as it is. So however much you apply the antibiotic to this, the resistance will give the defense against the antibiotic and help the bacteria to grow. So growth of bacteria on ampicillin medium suggests that all of these colonies have grown. But ಇಲ್ಲಿ ಕನ್ಫ್ಯೂಷನ್ ಏನಾಗುತ್ತೆ, 4 ಕಾಲನೀಸ್ ಒಳಗಡೆ ವಿಚ್ ಈಸ್ ರಿಕಾಮಿನೆಂಟ್ ಆ್ಯಂಡ್ ವಿಚ್ ಈಸ್ ನಾನ್ ರಿಕಾಮಿನೆಂಟ್ ಇದು ಗೊತ್ತಾಗಲ್ಲ. ವಿ ಕೆನಾಟ್ ಡಿಫ್ರೆನ್ಶಿಯೇಟ್. ಸೊ ಆ ಡಿಫ್ರೆನ್ಶಿಯೇಟ್ ಮಾಡಬೇಕಂತಂದರೆ ಗ್ರೋ ದ ಸೇಮ್ ಥಿಂಗ್ ಆನ್ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ಕಂಟೈನಿಂಗ್ ಮೀಡಿಯಮ್. Now for example, 3 colonies ಒಳಗಡೆ only 1 colony grew and other 3 colonies did not. ಸೊ ನಾವು ಈಗ ಎರಡು ಇದನ್ನು ಕಂಪೇರ್ ಮಾಡಿದ್ರೆ ಇದು ಒಂದೇ ಕಾಲನಿ ಗ್ರೋ ಆಗ್ತಾ ಇದೆ, ವಿಚ್ ಮೀನ್ಸ್ ದ ಸೆಲ್ಸ್ ಪ್ರೆಸೆಂಟ್ ಇನ್ ದಿಸ್ ಕಾಲನಿ ಹ್ಯಾಸ್ ನಾಟ್, ಲಿಸನ್ ಟು ಮೀ, ಹ್ಯಾಸ್ ನಾಟ್ ಲಾಸ್ಟ್ ದ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್. ದೇರ್ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ಈಸ್ ಇಂಟ್ಯಾಕ್ಟ್. Why? Because this medium had a tetracycline antibiotic. ಆ ಆಂಟಿಬಯೋಟಿಕ್ ಏನು ಮಾಡಬೇಕು? ಬ್ಯಾಕ್ಟೀರಿಯಾ ಸೆಲ್ಸನ್ನು ಕಿಲ್ ಮಾಡಬೇಕು, ಕರೆಕ್ಟ್? ಬಟ್ ಪ್ಲಾಸ್ಮಿಡ್ಸಲ್ಲಿ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ರೆಸಿಸ್ಟೆಂಟ್ ಜೀನ್ ಇಂಟ್ಯಾಕ್ಟ್ ಮೀನ್ಸ್ ನಾಟ್ ಡಿಸ್ಟರ್ಬ್ಡ್. ಇಂಟ್ಯಾಕ್ಟ್ ಇತ್ತಂತಂದರೆ ದ ಕಾಲನೀಸ್ ವಿಲ್ ಗ್ರೋ. ಬಟ್ ಕಾಲನೀಸ್ ಗ್ರೋ ಇದೆ ಆ್ಯಂಡ್ ಜೀನ್ ಇಂಟ್ಯಾಕ್ಟ್ ಇದೆ, ವಿಚ್ ಮೀನ್ಸ್ my experiment has failed. The gene of interest was not successfully inserted. ಅದೇ ಈ 3 ಕಾಲನಿ ಕಂಪೇರ್ ಮಾಡಿದರೆ 1, 2, 3 ದೀಸ್ ಆರ್ ನಾಟ್ ಗ್ರೋಯಿಂಗ್. ಆಂಪಿಸಿಲಿನ್ ಮೇಲೆ ಗ್ರೋ ಆಯಿತು ಬಿಕಾಸ್ ಆಂಪಿಸಿಲಿನ್ ನಾನು ಏನೂ ಮಾಡೇ ಇಲ್ಲ. ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ಮೇಲೆ ಗ್ರೋ ಆಗಿಲ್ಲ. ದಿಸ್ ಈಸ್ ಅ ಇಂಡಿಕೇಶನ್ ದ್ಯಾಟ್ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ರೆಸಿಸ್ಟೆಂಟ್ ಜೀನ್ ಈಸ್ ಟ್ಯಾಂಪರ್ಡ್ ಆರ್ ಅಲ್ಲಿ ಏನೋ ಒಂದು ಬೇರೆ ಡಿಸ್ಟರ್ಪ್ಷನ್ ಆಗಿದೆ, ವಿಚ್ ಮೀನ್ಸ್ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ಜೀನ್ ಇಸ್ ಬಿನ್ ಕಟ್ ಮಾಡಲ್ಲಿ ಏನು ಹಾಕಿದ್ವಿ ನಾವು, ವಿ ಹ್ಯಾಡ್ ಪುಟ್ ಅವರ್ ಜೀನ್ ಆಫ್ ಇಂಟ್ರೆಸ್ಟ್. ಸೊ ದ್ಯಾಟ್ ಮೀನ್ಸ್ ದಟ್ ದೀಸ್ ಆರ್ ಕಾಲ್ಡ್ ಆಸ್ ಟ್ರಾನ್ಸ್ಫಾರ್ಮೆಂಟ್ಸ್, or we can say recombinants. And that's how you select recombinants and non-recombinants, or transformants and non-transformants. Understood? Not understood? Any say something. Yes, no, what? No? Why so silence, people? This is scary. Either you have understood whole or understood nothing.
Next. Ampicillin medium.
Huh? Yes? Who is speaking again? Which class is speaking? Speak it louder. I could not hear. What are you telling? Which one you want?
Ampicillin.
ಹಾಂ, ಮುಂದೇನು ಆಂಪಿಸಿಲಿನ್ ಒಂದೇ ಮಾತು ಕೇಳಿದ್ರೆ ಮುಂದೇನು? ವಾಟ್ ಡು ಯು ವಾಂಟ್ ಎಕ್ಸಾಕ್ಟ್ಲಿ? ಟೆಲ್ ಮೀ. ವಿಚ್ ಕ್ಲಾಸ್ ಆರ್ ಯು ಫ್ರಮ್? ವಿಚ್ ಕ್ಲಾಸ್ ಇಸ್ ದಿಸ್ ಹ್ಯಾವಿಂಗ್ ಡೌಟ್? ವೇರ್ ಆರ್ ಯು ಸ್ಪೀಕಿಂಗ್? ಐ ಆಮ್ ನಾಟ್ ಏಬಲ್ ಟು ಹಿಯರ್ ಯು, ಡಿಯರ್. ಪ್ಲೀಸ್ ಸ್ಪೀಕ್ ಇಟ್ ಅಗೇನ್. Okay, what is your doubt? Again, I'm not able to hear you clearly. Don't waste the time. Hurry up. Huh? Coordinator, please, whoever there it is, please try to help the student. I am not able to understand what is the doubt. Voice was not clear.
Ma'am.
Huh? Which class is this, first of all? Okay, what are you saying? I'm not understanding. What do you want me to tell?
Transformer.
ಇದನ್ನೇ ಹೇಳಿದ್ದೆ ಇಷ್ಟು ತನಕ ಕಥೆ ನಾನು. ಟ್ರಾನ್ಸ್ಫಾರ್ಮೆಂಟ್ಸ್ ಆ್ಯಂಡ್ ನಾನ್ ಟ್ರಾನ್ಸ್ಫಾರ್ಮೆಂಟ್ಸ್, ದ ಸೇಮ್ ಥಿಂಗ್ ಯುರ್ ಆಸ್ಕಿಂಗ್?
Yes, ma'am.
Okay, did you understand where do we put the gene of interest? ಎಲ್ಲಿವರೆಗೂ ಅರ್ಥ ಆಯಿತು? At least tell me what point, till what point you understood and from what point you forgot or you did not understand. This is a artificially synthesized plasmid. PBr322 is an artificially synthesized plasmid. That plasmid contains multiple cloning sites. Bere cloning sites ide. And few cloning sites are present in between the marker. These are called marker genes, the second criteria. Why they are called marker? Marker edakke use ಮಾಡ್ತೀರಿ ನೀವು? To highlight something, to select something. Like that here, antibiotic resistant marker are used to select between transformants and non-transformants. Now, how they are used to select? They are called as resistant gene because presence of ampicillin resistant gene gives resistance against ampicillin antibiotic. Similarly, presence of tetracycline resistant gene gives resistance against tetracyclic antibiotic. Resistance means even in the presence of antibiotic, still the cells will grow. Antibiotic ಕೆಲಸ ಏನು? To kill the bacteria. But bacteria ಗೆ ನಾವು ಒಂದು ಡಿಫೆನ್ಸ್ ಕೊಡ್ತಾ ಇದ್ದೇವೆ. We are trying to defend the antibiotic. So antibiotic idru no, still the cells are growing because they have something which is antibiotic against yenide, they have a gene. So if I am putting my gene of interest in this section, yaudadru you can use. Okay, it is not necessary you have to use tetracycline. You can use ampicillin also. So for example, if I use ampicillin as a marker, andre avaga I should use either PVU1 or PST1 as a restriction enzyme, because ಆ marker ಅಲ್ಲಿ these two are restriction sites. ಈ ಮಾರ್ಕರಲ್ಲಿ ಬ್ಯಾಮೆಚಲಿನ್ ಸ್ಯಾಲ್ವನ್ ಆರ್ ದ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಸೈಟ್. ಸೊ ಮಾರ್ಕರ್ ಮೇಲೆ ಯಾವ ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಸೈಟ್ ಇದೆ, ಯೂಸ್ ದ ಸೇಮ್ ಎಂಜೈಮ್. ಸೊ ಯೂಸಿಂಗ್ ದ ಸೇಮ್ ಎಂಜೈಮ್, ಮೇಕ್ ಅ ಕಟ್ ಹಿಯರ್ ಆ್ಯಂಡ್ ಪುಟ್ ಮೈ ಜೀನ್ ಆಫ್ ಇಂಟ್ರೆಸ್ಟ್. ಇದರಿಂದ ಏನಾಯಿತು, ದಿಸ್ ಹೋಲ್ ರೆಸಿಸ್ಟೆಂಟ್ ಜೀನ್ ಏನಿತ್ತು ಗಾಟ್ ಸ್ಪ್ಲಿಟ್, ಕಟ್ ಆಗಿಬಿಡ್ತು. ಸೊ ಇನ್ನು ಮೇಲೆ ಸಪೋಸ್ ಇದ್ರ ಮೇಲೆ ನಾನು ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ಆಂಟಿಬಯೋಟಿಕ್ ಹಾಕಿದರೆ the cells will not be able to show resistance, they will become sensitive. Sensitive means they will die simply. Die ಆಗತ್ತೆ ಅಂದರೆ they will not grow. ಈಗ ಇದನ್ನ ಚೆಕ್ ಹೆಂಗೆ ಮಾಡ್ತೀರಾ? You cannot see bacteria by your eyes, you have to check it. How will you check? Use two plates, two Petri plates. On one plate suppose you are using ampicillin, ಅಂದರೆ ಆ ಪ್ಲೇಟಲ್ಲಿ ನಾನು ಆ ಮೀಡಿಯಮಲ್ಲಿ I have put ampicillin as an antibiotic and put this sample. Now in this sample I have a plasmid. This ide plasmid heng kanutte ega? How will you see this plasmid? This is the plasmid. Here I have tetracycline and alli nanu gene of interest hakbitte. So ampicillin is as it is, but tetracycline is broken. Now when I put this whole on the ampicillin containing medium, one sample alli multiple cells irutte, when I put that onto this medium, we can see many colonies. Illi bare 4 torsidini, there can be many colonies also. I have just shown you for example. Let's say there are 4 colonies growing. Idar artha enu? What will I conclude? That all these 4 colonies have ampicillin as a intact. Ee gene ge enu disturbance madilla. So all of them are growing. But idrinda recombinant yaudu, non-recombinant yaudu anta gottagalla. We cannot differentiate. Why? Because I have used tetracycline as a marker, or I have used BAM-H1 as a cloning site. So BAM-H1 ellide? Tetracycline alli ide. Suppose ನಾನು PVU as a restriction site use ಮಾಡಿದ್ರೆ ಆವಾಗ ಏನಾಗ್ತಿತ್ತು, ಆಂಪಿಸಿಲಿನ್ ಮೇಲೆ ಎಲ್ಲ ಗ್ರೋ ಆಗ್ತಿರ್ಲಿಲ್ಲ, ಯಾವುದೋ ಒಂದು ಗ್ರೋ ಆಗ್ತಿತ್ತು ಅಷ್ಟೇ. But now I have used what? I have used BAM-H1. BAM-H1 restriction enzyme use ಮಾಡಿದಾಗ it will cut the DNA. ಪ್ಲಾಸ್ಮಿಡ್ ಅನ್ನ ಇಲ್ಲಿ ಕಟ್ ಮಾಡುತ್ತೆ. ಕಟ್ ಮಾಡಿದ್ಮೇಲೆ I will put my gene of interest here and make a recombinant DNA. Then this sample, when I put on tetracycline containing medium, second set of experiment, idu bere experiment, idu bere, but madodu at a one time. Put 2 plates. One plate mele ampicillin, one plate mele tetracycline. Ampicillin mele ella grow agatte because we have not touched ampicillin gene. Tetracycline mele yen problem aayto? I have cut the tetracycline gene. Cut ಮಾಡಿ ಅಲ್ಲಿ ನನ್ನ gene ಹಾಕಿದ್ದೀನಿ, which means if this genes are becoming sensitive andre tetracycline illo antibiotic medium alli the cells will not be able to grow. Why? Resistant kodlikke resistant gene illa alli. ಆ resistant gene ಗೆ disturb ಮಾಡಿದೆ ನಾನು by putting my gene of interest. So when I compare this plate and this plate, avaga these 3 colonies ampicillin medium ಮೇಲೆ ಇತ್ತು but tetracycline ಮೇಲೆ ಇಲ್ಲ. This is a signal, or this is a identification that these 3 are transformants. They have successfully taken the gene of interest. ಅದಕ್ಕೆ ಈ resistant gene disturb ಆಯಿತು and they become sensitive. Now understanding, this is complicated, I know. ಅದಕ್ಕೆ ಮುಂಚಿಂದೆಲ್ಲ ಕಾನ್ಸೆಪ್ಟ್ಸ್ ಕ್ಲಿಯರ್ ಇರಬೇಕು. Understanding this?
Yes, ma'am.
Are you sure? Read this once a day. Odko read na. You cannot suddenly come and sit for the do. No, biotech is not that easy, people. You have to be very, very cautious. Iga, this is called as what? E method ige insertion inactivation anta ಹೇಳ್ತೀವಿ. This is called insertion inactivation or insertional inactivation. Artha ಏನು? What do you mean in English? Insert. See, ಈ ಪ್ಲಾಸ್ಮಿಡ್ ಅಲ್ಲಿ there were 2 markers. ಇಲ್ಲಿ ಒಂದು ಮಾರ್ಕರ್ ಇತ್ತು, ಇಲ್ಲಿ ಒಂದು ಮಾರ್ಕರ್ ಇತ್ತು. Okay, this is ampicillin marker, this is tetracycline marker. What I did, ಈ ಮಾರ್ಕರ್ ಮೇಲೆ ಇಲ್ಲೋ ಯಾವುದೋ ಒಂದು ರೆಸ್ಟ್ರಿಕ್ಷನ್ ಎನ್ಸೈಮ್ ತೊಗೊಂಡು ಇಲ್ಲಿ ಕಟ್ ಮಾಡಿದೆ. ಕಟ್ ಮಾಡಿದ್ರೆ ಏನಾಯಿತು? ಓ ಗಾಡ್ ಎಲ್ಲನೇ ಹೋಯ್ತು. ವೇಟ್, I cannot rub this much, not possible. Okay, now assume I have put small gene here. ಇದರಿಂದ ಏನಾಗುತ್ತೆ ಈಗ? ಸಪೋಸ್ ನಾನು ಆಂಟಿಬಯೋಟಿಕ್ ಇದರ ಮೇಲೆ ಹಾಕಿದ್ರೆ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ಜೀನ್ ಇಲ್ಲ, ಐ ಹ್ಯಾವ್ ಡಿಸ್ಟರ್ಬ್ಡ್ ಇಟ್. ಸೊ ದಿಸ್ ವಿಲ್ ಬಿಕಮ್ ಸೆನ್ಸಿಟಿವ್, ವಿಚ್ ಮೀನ್ಸ್ ಬಿಕಾಸ್ ಆಫ್ ಇನ್ಸರ್ಟಿಂಗ್ the gene of interest, the tetracycline resistant gene got inactivated. Inactivate ಆಯಿತು, ಅದಕ್ಕೆ therefore cells got sensitive to tetracycline. And therefore medium ಅಲ್ಲಿ ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ಇತ್ತಂತಂದರೆ, ದಿಸ್ ಇಸ್ ಅ ಪ್ಲೇಟ್, ದಿಸ್ ಇಸ್ ಅ ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಪ್ಲೇಟ್, ಟೆಟ್ರಾಸೈಕ್ಲಿನ್ ಇತ್ತಂತಂದರೆ ಕಾಲನೀಸ್ ವಿಲ್ ನಾಟ್ ಗ್ರೋ. ಸೊ ಇನ್ಸರ್ಷನಲ್ inactivation means because of inserting our gene of interest, the gene got inactivated. This can be used for this also, anything is fine. Same thing can be used by lac operon. If you remember lac operon ali, suppose this is a lac Z gene. So instead of using antibiotics, use the enzyme gene. So enzyme gene iddaga, suppose ನಾನು ಇಲ್ಲೇ ನನ್ನ ಇದನ್ನು ಹಾಕ್ಬಿಟ್ಟೆ, ವಾಟ್ ಐ ಪುಟ್ ಹಿಯರ್ ಜೀನ್ ಆಫ್ ಇಂಟ್ರೆಸ್ಟ್. ಈಗ ಇದರಿಂದ ಏನಾಯಿತು? ವಾಟ್ ಈಸ್ ದ ಕಾನ್ಸಿಕ್ವೆನ್ಸ್? Z gene should make beta galactosidase, and beta galactosidase enzyme will break lactose into glucose and fructose. Bro, this is a molecular inheritance, you have forgotten. So now this Z gene alli, what did I do? I put gene of interest. So Z gene disturbed, no Z gene, no beta galactosidase, no beta galactosidase, no lactose used, ಯಾವುದು ಪ್ರೊಸೆಸ್ ಆಗಲ್ಲ. ಸೊ ಆ್ಯಂಡ್ ಇದಕ್ಕೆ ಏನು ಮಾಡ್ತೀವಿ? ಟು ಕ್ರಾಸ್ ಚೆಕ್, ಅಲ್ಲಿ ಏನು ಮಾಡಿದ್ವಿ? ಹಿಯರ್ ವಾಟ್ ವಿ ಡಿಡ್, ವಿ ಯೂಸ್ ದ ಆ್ಯಂಟಿಬಯೋಟಿಕ್. ಪರ್ಪಸ್ಲಿ ಆ ಮೀಡಿಯಮಲ್ಲಿ ನಾನು ಆ್ಯಂಟಿಬಯೋಟಿಕ್ ಹಾಕಿದ್ವಿ. ಇಲ್ಲೇನು ಮಾಡ್ತೀವಿ? ಬಿಕಾಸ್ ವಿ ಆರ್ ಯೂಸಿಂಗ್ ಎನ್ಸೈಮ್, ವಿ ವಿಲ್ ಯೂಸ್ ಕ್ರೋಮೋಜೆನಿಕ್ ಸಬ್ಸ್ಟ್ರೇಟ್. Chromogenic substrate means color giving substrate. ಈಗ ಲ್ಯಾಕ್ ಝೆಡ್ ಜೀನ್ ಬೀಟಾ ಗ್ಯಾಲಕ್ಟೋಸಿಡೇಸ್ ಸಬ್ಸ್ಟ್ರೇಟ್ ಯಾರು? ಲ್ಯಾಕ್ಟೋಸ್. ಸೊ ಕಲರ್ ಗಿವಿಂಗ್ ಲ್ಯಾಕ್ಟೋಸ್, ದಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಅಲೋಲ್ಯಾಕ್ಟೋಸ್ ಅಂತ ಒಂದು ಸಬ್ಸ್ಟ್ರೇಟ್ ಬರುತ್ತೆ. ದ್ಯಾಟ್ ಸಬ್ಸ್ಟ್ರೇಟ್, ಕಲರ್ ಗಿವಿಂಗ್ ಸಬ್ಸ್ಟ್ರೇಟ್, ಐ ವಿಲ್ ಪುಟ್ ಇನ್ ದ ಮೀಡಿಯಮ್. ಈಗ ಆ ಮೀಡಿಯಮಲ್ಲಿ ಸಬ್ಸ್ಟ್ರೇಟ್ ಹಾಕಿದ್ದೀನಿ. ಸಪೋಸ್ ದಟ್ ಜೀನ್ ಈಸ್ ಇಂಟ್ಯಾಕ್ಟ್, ಅಂದರೆ ಆ ಜೀನಿಗೆ ಏನೂ ನಾನು ಡಿಸ್ಟರ್ಬ್ ಮಾಡಿಲ್ಲ ಅಂದರೆ it will produce lactose and disturb the lactose into glucose and fructose. And because the substrate is color giving, you will find blue colonies. But blue colonies antandre, they are non-transformants. ಈಗ suppose this gene is intact, ಅಂದರೆ ಈ ಜೀನಿಗೆ ನಾನು ಏನು ಮಾಡಿಲ್ಲ, this gene is as it is. ಸೊ ದೀಸ್ ಜೀನ್ ಇಸ್ ಆಸ್ ಇಟ್ ಇಸ್ ಅಂತಂದರೆ ಇದ್ರ ಜೊತೆ ಸಬ್ಸ್ಟ್ರೇಟ್ ಏನಿದೆ? ಐ ಹ್ಯಾವ್ ಅ ಕಲರ್ ಗಿವಿಂಗ್ ಸಬ್ಸ್ಟ್ರೇಟ್, ಯಾವುದೋ ಒಂದು ಕಲರ್ ಗಿವಿಂಗ್ ಸಬ್ಸ್ಟ್ರೇಟ್ ಇದೆ. ಕಲರ್ ಗಿವಿಂಗ್ ಅಂದರೆ ಏನು? ವೆನ್ ದಿಸ್ ಎನ್ಸೈಮ್ ವಿಲ್ ಆ್ಯಕ್ಟ್ ಅಪಾನ್ ದಿಸ್ ಸಬ್ಸ್ಟ್ರೇಟ್, ಇಟ್ ವಿಲ್ ಪ್ರೊಡ್ಯೂಸ್ ಅ ಕಲರ್, ಬ್ಲೂ ಕಲರ್. ಸೊ ಜೀನಿಂದ ಎನ್ಸೈಮಿಂದ ಕಲರ್, ವಿಚ್ ಮೀನ್ಸ್ ಆ ಜೀನಿಗೇನು ಡಿಸ್ಟರ್ಬೆನ್ಸ್ ಆಗಿಲ್ಲ, ಆ್ಯಂಡ್ ದೇ ಆರ್ ನಾನ್ ಟ್ರಾನ್ಸ್ಫಾರ್ಮೆಂಟ್ಸ್. ಸಪೋಸ್ ಜೀನ್ ಡಿಸ್ಟರ್ಬ್ ಆಗಿದೆ, case 2, suppose gene is disturbed, then there is no enzyme. If no enzyme, then substrate is not converted, and substrate convert agilla andre nimage yavadu color sigalla. So you get white colonies, and white colonies are called as transformants. or recombinants. So this is called blue-white screening or blue and white colonies. Two methods to select whether it's a recombinant or non-recombinant. These are the two methods. One by using selectable marker of antibiotic resistant gene, one by using the chromogenic substrate. ಯಾವುದಾದ್ರೂ ಯೂಸ್ ಮಾಡ್ಬೋದು ಇನ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್. ದ ಫಸ್ಟ್ ಆಂಟಿಬಯೋಟಿಕ್ ರೆಸಿಸ್ಟೆಂಟ್ ಜೀನ್ ಇಸ್ ಲಿಟಲ್ ಬಿಟ್ ಕ್ಯೂಬರ್ಸಮ್ ಮೀನ್ಸ್ ಡಿಫಿಕಲ್ಟ್, ಯಾಕಂದರೆ ಆಟ್ ಅ ಟೈಮ್ ಯು ಹ್ಯಾವ್ ಟು ಮೇಕ್ ಟು ಪ್ಲೇಟ್ಸ್. ಸೊ ದಟ್ ಫಾರ್ ದಿಸ್ ಈಸ್ ಈಸಿ. ಒಂದೇ ಪ್ಲಾಸ್ಮಿಡ್ ತೊಗೊಳೋದು ವಿಚ್ ಹ್ಯಾಸ್ ಅ ಲ್ಯಾಕ್ ಒಪೆರಾನ್ ಇರೋ ಪ್ಲಾಸ್ಮಿಡ್ ತೊಗೊಂಡ್ಬಿಟ್ಟು ಆ್ಯಡ್ ಅ ಕಲರ್ ಸಬ್ಸ್ಟ್ರೇಟ್, ಎನಿ ಕಲರ್ ಕ್ರೋಮೋಜೆನಿಕ್ ಸಬ್ಸ್ಟ್ರೇಟ್, ಜೀನ್ ಎಂಝೈಮ್ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದರೆ enzyme will give you blue color. Enzyme produce ಮಾಡಲಿಲ್ಲ ಅಂತಂದರೆ it will give you white color. ಅಷ್ಟೇ, easy. So blue colors are non-transformants and white color are transformants. This is difficult, I know. ಇದನ್ನು ಮೊದಲು ನಿಮಗೆ ಕಾಲೇಜಲ್ಲಿನೂ ಕಲಿಸಾಗಿದೆ, ಆವಾಗೂ ನೀವು ಓದಲಿಲ್ಲ. ಇವಾಗ ನಾನು ಹೇಳ್ತಾ ಇರೋದು many of you Rana girls were not attentive. I don't know what you're going to do because I cannot keep on repeating this. Read and come for tomorrow's class. I'm going to stop here. We will continue with solving questions on this and the next concepts. Read and come. Illa arth agalla. I'm telling you 100 times. Read and come. Okay? Chalo, we'll meet you tomorrow, people. Ansate odkondu balli mundindella. Okay? Have a good day. Bye bye.
Yes.
Thank you, ma'am. Thank you, ma'am.